Okay, so the second stage of photosynthesis, it used to be called the dark reactions when I was going through school. This is what they told us that they were called because they used to think that they only happened in the dark, in the absence of sunlight. So we know that the first stage of photosynthesis for all plants, they need sunlight in order to initiate the entire process of photosynthesis. But now what they're realizing is that stage two, it can happen in the dark, but it doesn't necessarily always happen in the dark. So they've renamed this stage as the light independent reactions, meaning that it can happen without light, but it can also happen in the presence of light. So it doesn't need light in order for it to happen. And the scientist who discovered all of this was a scientist by the name of Calvin. So it also has the name the Calvin cycle. And so the three main points for this stage. Now this stage resembles very close to the things that happen during our Krebs cycle. But we're not going to go into as much detail as we did with our cellular respiration. I'm going to focus in on what I want you to know out of this cycle. So the first thing you should know is that they don't necessarily require light, but they can happen in the dark if they need to. The reactions are very important because they fix inorganic carbon in the form of carbon dioxide to organic carbon in the form of glucose. And the reason that they call this carbon fixation is because of this step here. So there's the Calvin cycle is broken down into three stages. One of them we call carbon fixation because we're basically taking carbon dioxide from the atmosphere and we're making it into glucose. And then the final thing I want you to know is that it happens in the stroma of our chloroplasts. So that's much like the matrix of our mitochondria. So try not to we're not going to fill in all these blanks because it's a lot of words we don't need to know and a lot of steps we also don't need to know. So we're going to ignore this, and what I want you to focus on is really just three main things, which I'm going to show you. Okay, so let's put it in purple what I want you to know. What I want you to know is that the Calvin cycle starts off with three, and they're not very clear here, but there's three carbon dioxide molecules from the atmosphere entering the Calvin cycle, which we saw in the big picture diagram that I gave you. There's also three water entering here. So this marks the beginning part of our cycle. Remember in Krebs cycle we had a complete cycle where we regenerated our starting materials? The exact same thing's happening here in our Calvin cycle. We have the entry of carbon dioxide from our atmosphere, and it's going to join with this. So this is our first stage. And this molecule here has a really, really long name. It's called ribulose bisphosphate. I don't need you to know that whole name, but they do have a short form. It's called RUBP. And bisphosphate or biphosphate just means there's two phosphates on the end. So this is a phosphate and this is a phosphate. Remember the importance of transferring phosphates from ATP to ADP. The same thing's happening throughout. So anytime you see these circles here, it's phosphates that are coming and going, being added or removed. And so this is stage one here. This molecule enters with carbon dioxide, and we end up in our first step making a 3-carbon molecule with a phosphate attached to it. Okay, and here we have some ATP being broken down into ADP plus PI. And remember that we're getting some energy when we do that sort of conversion, when we've got stored energy in our bonds of ATP, and when we release those, we're releasing energy for some of these reactions to happen. And one of them is that we add an extra phosphate on. So this guy just had one phosphate, it now has 2 phosphates at the end. And so we can mainly break this this cycle down into 3 main phases, I guess you could say. So in the first phase, we're gonna call this carbon fixation. And that's because we took carbon dioxide from the atmosphere and we fixed it or converted it into a carbon molecule, an organic carbon molecule. Okay, and then a few stages happen here, and I'll just fill these in so you can see what's happening. We have NADPH being converted into NADP positive. Remember, all this reaction here is it's transferring its electrons. So its electrons that are stored in hydrogen are being oxidized. So NADPH is oxidized because it's losing its electrons from hydrogen. And remember, in any type of redox reaction, if one molecule is losing electrons, another one's gaining them. And that's what's happening here from this molecule to this molecule, which is why we call this the reduction phase. And so remember, don't be concerned with the names of all the molecules or what their structures look like, just what I'm writing on this diagram here. That from this molecule to this molecule, we're reducing something. And it's the exit here of where we're going to get our sugar. So this molecule here is called glyceraldehyde 3-phosphate. So we're going to just call it, and all you need to know is G3P. And G3P is a 3-carbon sugar that has phosphate attached to its 3rd carbon, which is why we end up with the name G3P. From this production of G3P, we get our glucose. And basically the way that we get our glucose is through the reverse steps of glycolysis. So remember the process of glycolysis that we talked about during cellular respiration? It's basically 10 steps, starting with glucose, and we were breaking glucose down, and we ended up forming glyceraldehyde 3-phosphate. So in plants, through the first few steps of the Calvin cycle, we're making glyceraldehyde 3-phosphate, which was one of the stages of glycolysis. And basically what they do is they just reverse the stages of glycolysis in order to make the glucose molecule that we start with. And that's basically what happens here. So we end up, if I can recap what's happened so far, carbon dioxide comes in from the atmosphere. It combines with RuBP. We call that the carbon fixation step. A few steps happen here where we use up some energy from ATP. We have a redox reaction with NADP, and we call that the reduction phase. Our product is glyceraldehyde 3-phosphate, and from glyceraldehyde 3-phosphate to glucose, I'm going to extend this a bit, we make glucose from reverse steps of glycolysis. Okay, but this cycle still continues, and the reason it continues is because we want to regenerate this molecule that we started with, just like we did in the Krebs cycle. And so this side of the cycle is just called, or the phase is just called, the regeneration of RuBP. And that's it. That is all I want you to know from this. Actually, just these two things here. We have a little bit more water coming in over here, and we have ATP being broken down into ADP plus phosphate. Okay, so be familiar with the 3 phases of the Calvin cycle. The product that comes out, G3P is all you need to call it, that undergoes reverse glycolysis to form our glucose molecule, because that's the one product we're looking for out of photosynthesis. And then the final stage is we just want to make our RuBP, which is a starting material for this cycle to continue over and over again.